Hello and welcome back to the deep dive on the hallmarks of cancer. And today we're going to be talking about tissue invasion and metastasis of the cells. So, this is one of the most deadly capabilities of cancer cells, and it's a really, really bad sign. And it basically means that cancer cells are allowed to spread from the original site and establish new tumors in distinct organs. And it's responsible for 90% of cancer related deaths because when a tumor is localized, it's relatively easier to treat, might it be through surgery or through other methods, but when it's spread through other tissues, it becomes far more difficult to target the treatment and the therapy because it could be literally anywhere within the body, and there are methods to detect it, but it can continue to spread like a virus spreading along a population. So today we're going to break down the molecular mechanisms behind this and hopefully use some analogies and some new methods of explanation to make it easier to understand. So the first thing we're going to talk about is cell adhesion molecules, or CAMs. And these proteins are essential for maintaining the structure of tissues by facilitating cell to cell and cell to ECM interactions, where ECL stands for extracellular matrix. So first, let me talk about the etymology of the word, because I find that it often helps to remember the name of something if you remember why it's named that way. So catherines are named for calcium-dependent adhesion, and that explains everything except the ending of ins in the word, like cather and then ins, right? But the ins comes from adherent junction, which are protein complexes that occur at cell-to-cell junctions in epithelial and endothelial tissues. Now, when you hear catherines, you'll always associate the connections, the junctions between the cells, or at least I hope that's what this serves. Another unique benefit of understanding the name is that it can also carry clues regarding how it functions. So, occasionally scientists can be pretty descriptive, just not in the language we understand and in a pretty weird way. So the first clue about how these catherines work is through calcium ions, but there's obviously more to that explanation. So firstly, when the catherine proteins are produced, they travel to the cell membrane, and it establishes itself as a transmembrane protein with distinct cytoplasmic and extracellular domains. The extracellular domains consist of 5 catherine repeats, and each repeat is made up of around 120 amino acids, I believe. And when calcium ions come in, they stabilize these catherines, which I don't really understand what it means, but they bind to specific areas and they react to the aspartic and glutamic acid to make them more rigid. And this allows for the forming of dimers of catherines, where they can bind to catherines that are on other cells like a zipper and create sort of trans dimers which hold the cells together. You take away calcium from the system and the cells won't be held together anymore because the catherines won't work. But another thing that makes these catherines work, or the binding function, is that what is the inside binder to? The cytoplasmic domain. The cytoplasmic domain is made up of 3 catenins, alpha, beta, and gamma, which are all linked to the actin cytoskeleton of the cell. If there are no catenins, this linking does not happen. And what we're talking about here is E-catherine, which stands for epithelial catherine. And other type of catherines exist, such as N-catherines, which stand for neural catherines, and P-catherines, which stands for placental catherines. But now that the function is clear, what does this have to do with cancer? So firstly, there could be gene mutations in the CDH1 gene, which encodes the E-catherine, and it can alter the protein structure, which will impair its ability to form stable interactions with neighboring cells. This can occur through missense mutations, and missense mutations basically means changing an amino acid, or it can happen through truncating mutations, which is when you shorten a protein. There's also transcriptional repression, where transcriptional repressors can bind to the CDH1 promoter, reducing E-catherine expression. DNA methylation and histone modifications are epigenetic changes that can also silence CDH1 expression. And DNA methylation, the upregulation of this activity, is commonly associated with a lot of type of cancers with really poor prognosis. And then we have proteolysis, where proteolytic enzymes like matrix metalloproteinases or ADAM10 can cleave E-catherine, leading to the release of its extracellular domain. And if the main linking part is gone, if the zip is gone, nothing's going to hold it together anymore. And this process disrupts cell-to-cell adhesion and allows cells to become more motile. And the loss of E-catherine disrupts anti-growth signaling pathways that normally prevent cells from proliferating excessively or detaching. And this loss will promote cellular motility and invasiveness. And when we don't want this to happen, it creates really bad effect. So just to conclude this explanation with an analogy, imagine a group of people forming a human chain to hold up a large barrier. Each gathering acts like the strong grip between each other that they have on the strong hands, keeping the barrier intact. But if some individuals start letting go or weakening their grips, which can happen due to E-cadherin mutations, repression, or breakdown, the barrier will become unstable. And this instability creates gaps through which individuals, which we can consider to be the cancer cells, slip out and move away from the group, thus invading new areas. The next part we're going to talk about is NCAM, and NCAM stands for neural cell adhesion molecule. Sorry for not writing that in the PPT, but NCAMs belong to the immunoglobulin superfamily and have the following structural features. Also, immunoglobulin can be shortened to IG, so I'm just going to refer to it as IG for now. I'm not going to say immunoglobulin. I guess we'll see, but it's good to know the shortened form. So now I'm going to explain the structural features of the immunoglobulin family. So there's an extracellular domain which contains the immunoglobulin-like domains and fibroconnectin type 3, which is FN3 repeats. These domains are involved in cell-to-cell interactions and provide the structural basis for adhesion. They also have the transmembranous domain, which anchors the NCAM in the cell membrane, and then they have the intracellular domain, which links the intracellular signaling pathways in the cytoskeleton. Although it is less well-defined compared to other adhesion molecules like cadherins, it's still being investigated. So for the mechanism of NCAM adhesion is that there's homophilic bonding, which means that NCAM molecules on one cell bind to identical NCAM molecules on the adjacent cell. And this interaction is similar to cadherin-mediated adhesion, but it involves different structural motifs. Then we also have the immunoglobulin domains, and the extracellular immunoglobulin-like domains of the NCAMs engage in adhesive interactions, providing both specificity and specificity, I think, and strength to the adhesion between cells. And then you have the alternative splicing and isoforms. And NCAMs can exist in several isoforms due to the alternative splicing of the mRNA. And these isoforms differ in number and type of extracellular domains and can influence the adhesive properties and functional roles of NCAMs. And different isomers may have varying adhesive strength and interactions with other cell surfaces, and it may affect processes like neurodevelopment and synaptic plasticity. So you see a lot of linking, or especially in this part, you see a lot of linking between neuroscience, another up-and-coming field in bio. So, well, not up-and-coming, it's been established for a really long time, but it's gaining increasing interest and importance. So the role of NCAMs in neural development is that during development, NCAMs are crucial for guiding neural cell migration and positioning, and they help establish the correct connections between neurons and other cells. And NCAMs also facilitate the migration of neural progenitor cells and newly differentiated neurons by mediating cell-to-cell adhesion and interactions with the extracellular matrix. They also play a role in recognizing and establishing connections between neurons. And for the regulation of NCAM function is the one is post-translation modifications. And one example is glycosylation, where NCAMs undergo extensive glycosylation, which can affect the adhesive properties and interactions, and changes in the patterns of these can affect the binding affinity and function of these NCAMs. And we also have isoform switching, which we described above, where different NCAM isoforms can be either upregulated or downregulated, which can have an impact on cell adhesion and signaling processes. But what is the impact of cancer-related alterations? Now, in cancer, NCAM can switch from more adhesive forms to less adhesive forms, and this change can result from alternative splicing or post-translation modifications such as glycosylation, which affect the adhesive properties of NCAMs. And the switch to less adhesive forms would reduce the strength of cell-to-cell adhesion and could facilitate the detachment and migration of cancer cells. And this can promote their ability, as we learned above, to spread the primary tumor to other tissues. And less adhesive isoforms can also lead to less cell-to-cell contact, which enhances cell motility and invasiveness. So just as an analogy to summarize this, consider NCAMs as a handshake. So a firm handshake keeps the people firmly connected, and in cancer it's a looser grip, so people will easily slip away. It's a very similar analogy to I actually don't know why I said that. But the next part we're going to talk about is integrins. And integrins are named based on their function and characteristics, right? The name integrin is derived from the Latin word integer, which means whole or complete, which is also true in math. Integers are just whole numbers. And this is fitting because integrins bind cells to the extracellular matrix and integrate cells into their surroundings. And this name reflects their role in linking cells to the extracellular matrix and maintaining its structural integrity of tissues. So to understand integrins, let's talk about their structure and how they work. So the structure of integrins is, integrins are composed of two different subunits, the alpha and the beta, and the different types of these subunits. So integrins are named according to their specific alpha and beta combinations. And two examples at the top of my head would be the alpha 5 beta 1 or the AV alpha, I think it's alpha V, I'm not sure how to pronounce that, beta 3. And each integrin has a transmembrane domain that spans the cell membrane with extracellular and intracellular domains. So the extracellular domain of the integrins contains binding sites for ECM components. These sites interact with specific proteins like fibroconnectin, collagen, and laminin, and the binding of these integrins to the extracellular matrix induces conformational changes that allows for stable adhesion. Unlike cadherins, integrins don't rely on calcium ions for stabilization. Instead, their function depends on other mechanisms. Integrins can bind to extracellular matrix, I'm just going to say ECM, it's too long, ECM proteins, and this activation process involves the integrin shifting from a low affinity state to a high affinity state, enhancing its ability to bind to ECM components. So, and for the intracellular domain of integrins connects to the cytoskeleton through adaptor proteins such as talin and vinculin. This connection helps anchor the cell to the ECM and transduce mechanical signals. And they produce signaling pathways that regulate cell behavior, including migration, proliferation, and survival. So it's not just a physical thing, you can consider it to be like a behavioral thing. So these integrins are pretty important when it comes to metastases of cancer cells. And there's something called integrin switching, where in normal tissues, integrins bind to well-structured ECM components like fibroconnectin and laminin, and these interactions help maintain tissue structure and function. Now, degraded ECM binding refers to when in cancer cells, integrins can switch to different types that bind to degraded ECM components or components associated with the stroma. This allows cancer cells to interact with and invade through the altered ECM. And there are functional adaptations which are crucial for cancer cells to adapt to the new environment during invasion of metastases. For example, integrins like AV beta 6 are often upregulated in cancer and bind to ECM fragments, promoting invasion and metastases. As a quick analogy to visualize integrin function and switching, think of it like this. Imagine you're walking on a solid paved path and you wear sturdy boots that are ideal for stable ground. This is analogous to healthy integrins binding to healthy ECM components in normal tissues. Now think if you move to rugged, uneven territory, which represents degraded ECM, you'll need specialized hiking boots to navigate effectively. And this switching to different types of boots is like integrins changing and binding to new ECM components in cancer. And this is what helps the cells move, and this is what is established in new areas. And now we're going to talk about the extracellular proteases, which are named based on the function and location. The term protease comes from the Latin proteus, which means changing, and ase just means an enzyme. And these enzymes change proteins. These enzymes change proteins by breaking them down. And extracellular indicates that these proteins act outside the cell in the space between the cells where the ECM is located. So there are many different types of extracellular proteases. One is matrix metalloproteinases, which are known as MMPs, and these are a large family of zinc-dependent endopeptidases that degrade various ECM components, including collagen, elastin, and proteoglycans. And then we have the urokinase type plasminogen activators, which is UPA, and this enzyme converts plasminogen to plasmin, which then breaks down fibrin in blood clots and other ECM components. So proteases are initially produced as inactive forms called proenzymes or zymogens, and these proenzymes need to be activated to perform the function. And Activation typically involves proteolytic cleavage, where a portion of the proenzyme is removed, which enables a protease to adopt its active form. For example, pro-MMPs are converted into active MMPs by enzymes like tissue inhibitor of metalloproteinases, which is known as TIMP or TIMP, which is a pretty funny name. And once activated, these proteases break down ECM components by cleaving peptide bonds in proteins. This degradation disrupts the ECM structure, making it easier for cells to migrate through the tissue. And many proteases are located on the cell surface or secreted near the cell membrane, and this proximity enhances their ability to degrade the ECM directly in the area where it's needed. So, for protease function in cancer invasion, they could either be upregulated or downregulated. So we have increasing expression, where in the cancer cells the genes that encode the proteases are often upregulated, which causes an increased production of these enzymes, and this heightened production facilitates a more extensive ECM degradation. Simultaneously, protease inhibitors, which normally keep protease activity in check, are downregulated, and this allows greater protease activity and thus more ECM breakdown and increased expression, and it creates a vicious cycle. And proteases that are initially inactive are activated, which enable them to efficiently degrade the ECM. So, and then we have the ECM breakdown, wherein by degrading the ECM, proteases create pathways for cancer cells to invade surrounding tissues and enter blood vessels. And ECM degradation also contributes to angiogenesis, which is formation of new blood vessels, and facilitates the spread of cancer cells to other parts of the body. So now let's talk about what all cancer research eventually boils down to and why we do it, which is the treatment. So firstly, we have the treating of E-cadherins in cancer treatment. So in cancer, the E-cadherins are mutated, replaced, or cleaved, and that disrupts cell adhesion and promotes the motility and invasion. So what are the therapeutic approaches for this? Firstly, it's restoring E-cadherin function, which there's research that is going on to find a way to restore or enhance E-cadherin function to reestablish cell adhesion and prevent invasion. And this might involve gene therapy to correct mutations or small molecules that stabilize E-cadherin. The next method is to inhibit E-cadherin cleavage. So basically, everything that's over here is a reversal of the problems that cancer causes and a return to normal function. So by inhibiting E-cadherin cleavage, by preventing the breakdown of E-cadherin, these drugs aim to maintain cell adhesion and reduce cell motility. And they also use targeted transcriptional repressors, which are strategies to counteract transcriptional repression on E-cadherin, and use drugs that inhibit DNA methylation or histone deacetylation, which can help restore E-cadherin expression. For the targeting of NCAM, there are many therapeutical approaches. The first one is the modulating of NCAM isoforms. So researchers are exploring drugs that can alter NCAM isoform expression or function. For example, targeting specific isoforms that are less adhesive could reduce cancer cell motility. And some drugs that alter glycosylation patterns of NCAM might be used to modify its adhesive properties and impact cancer progression. For targeting integrins, the first approach is that drugs that inhibit integrins, for example, monoclonal antibodies or small molecules, are being developed to prevent cancer cells from binding to ECM components. And these inhibitors block the interaction between integrins and the ECM, and they disrupt cell adhesion and migration. They could also target specific integrins, with some therapies focusing on integrins that are upregulated in cancer, for example, alpha V beta 3. And by targeting these integrins, drugs can inhibit cancer cell invasion and metastases. And the final thing is the targeting of extracellular proteases. So drugs that inhibit proteases, for example, MMP inhibitors, are designed to prevent the breakdown of the ECM. By reducing ECM degradation, these inhibitors can limit cancer cell invasion and metastases. And by targeting activation pathways, researchers are currently trying to develop methods and strategies to block the activation of proteases from their inactive form, which can reduce their ability to degrade ECM components. So, in conclusion, tissue invasion and metastases are multi-layered processes that involve many molecules, And by understanding the roles of CAMs, integrins, and extracellular proteases and how they interact with the surrounding environment, we can create strategies, novel strategies that combat cancer spread. Thank you for joining today. So long, my mouth hurts. See you guys in episode 7.